This is Dr. Leonard Calabrese. I'm Professor of Medicine at the Cleveland Clinic Lerner College of Medicine and the R.J. Fazenmeyer Chair of Clinical Immunology at the Cleveland Clinic. This is the blue line, in which we'll discuss the extrapolation of indications and how biosimilars are named. We'll review the additional requirements that a biosimilar must meet to be considered interchangeable with its reference product. First, let's start out about this concept of extrapolation. In its simplest terms, extrapolation is just what the lexicon would suggest, that a biosimilar, once demonstrated to be physiochemically and immunochemically similar and have the appropriate pharmacokinetics and pharmacodynamics, then is demonstrated to be clinically efficacious and safe in a disease. If the originator drug is approved for multiple diseases, it is accepted by extrapolation that the approval in one disease will give it an approval in the other diseases, such as a biologic such as adalimumab, which is approved for many diseases, including rheumatoid arthritis, psoriasis, spondylitis, and more. If I have a biosimilar that is demonstrated to be clinically efficacious in psoriasis, I may be given by extrapolation FDA approval for the other indications. A second and somewhat confusing area in biosimilars is this naming process. Now, at first blush, this would seem to be an easy challenge, but think about it for a moment. So already we have biosimilars that have been FDA approved to several immunoactive drugs used to treat immune-mediated immunoinflammatory diseases, or IMIDs. Adalimumab is a good example. There are 2 approved adalimumab molecules. So how will we name them? Well, they will retain the name of the chemical compound of the originator, adalimumab, and then they'll be given an identifier. And the identifier is a random 4-letter suffix that is applied. So it may be ATTO or ABDM, or it could be XYAC, etc. When I first heard this, I was confused about this. I said, well, why isn't it named just for the company that developed it? The response was actually rather clarion. Over time, that company may be sold, or the product may be sold to another company. So at the moment, it's important to be able to uniquely identify it. We could envision 3 or 4 years from now that there may be 5 biosimilar adalimumabs and 6 biosimilar infliximabs. So the pharmacy will have to keep track of them. The provider will have to keep track of them, and ultimately, the patient will have to keep track of them. So we need unique identifiers. So what may be complex right now, I think will provide needed clarity as we move ahead. The next term, which causes concern for everyone who's trying to understand this field of biosimilars, is this notion of interchangeability. Interchangeability is an FDA designation. And as I will point out, it requires a different set of standards from clinical trials to be given this. And if a biologic was deemed interchangeable, the implications for clinicians and patients would be that the pharmacist, without pre-approval of the prescribing clinician, would be able to insert the biosimilar for the originator. So if I was using the originator infliximab and there is a biosimilar infliximab ABCD, that pharmacist would then merely insert this as they would a generic for a small molecule. This has raised a lot of concerns in the provider community. So what are those additional considerations that we have to think about? Well, shouldn't a product be deemed interchangeable? If I, the clinician, I would want to be notified for this. And indeed, implicit in this is notification, but that could be post hoc. Now, because we live in a federation, each state has its own laws, and states are now creating their own substitution laws in advance of interchangeability to regulate this. The pharmacist substitution will have to abide by state laws, and there will have to be a framework. It will have to have product criteria. The clinician can, just like we can for small molecules, write dispense as written. We have to have laws about how this will be communicated, how we will keep our records, and how health systems may be or may not be exempted from this. I'm showing you on this slide examples of enacted biosimilar substitution laws in multiple states, and these are now moving with great rapidity across the country, looking at what time will the pharmacist have to tell the clinician that they're substituting, what other provisos will be in this interchangeable designation. If I wanted to summarize the common elements of interchangeability, I would say these are the four tenets. The biologic product under consideration must first be designated as interchangeable. Now, I'm going to stop right here, and anyone listening to us right now, I will tell you there are no interchangeable biosimilars that have been approved thus far. There is only a single study that has been designed that is now being launched, so it'll be some time before this occurs. So we don't need to worry about it immediately, but it's something on our radar screen. So the second tenet is that the prescriber would be able to prevent substitution, just like we can for small molecules, by including the DAW, or dispense as written. The third common element is that this will not happen unbeknownst to us. The prescriber must be notified, and so some communication has to occur. And finally, and most importantly to me, the patient must be notified that a substitution or switch has been made. Now, all of this will be shaded by the state-to-state variations, which are working their way through legislative processes.
So in summary, an approved biosimilar can be expected to have the same efficacy and safety profile, which means no clinically meaningful differences, as the reference product for all approved indications. Based on the totality of evidence, including analytic studies, physical, chemical, immunochemical, and clinical studies, biosimilars can be approved to treat indications even without clinical trials in that indication. That is called extrapolation. Finally, interchangeability is a tall hurdle for biosimilar approval, and as of yet, there are no approved agents with this designation.